Hello students. Now today in this video we are going to learn about the polymerase chain reaction. We know that is a very interesting as well as a very vital technique in the biotechnology or various genetic operations. So let us study because this polymerase chain reaction, because in many a times when we are working with DNA, the DNA is not, we don't get DNA in sufficient amount, clear? So suppose for DNA fingerprinting, we need DNA. So there are only some blood samples, and that also only WBC can provide as DNA. Or we want to study, or we want to, in biotechnology, we want to incorporate some gene from one organism to other. Then also we are not getting a huge amount of DNA. Suppose we want to incorporate insulin gene into E. coli. So we are not getting a huge amount of insulin gene. Clear, we are getting it from certain human being, so that is a restricted amount we are getting. So but if we start working with that only, and then after a part, it may happen that the experiment may went wrong, or it may, there are several factors are there for a successful incorporation of gene. So we need to start with a lots of amount of that particular gene of interest. So when whatever the technique we are going to apply, so for that we need to amplify the gene of our interest, or that segment of DNA which we need for our further purposes. And this amplification is done by PCR. So PCR is a polymerase chain reaction. It is an in vitro, that means outside the cell the DNA synthesis occurs, clear? So it is an in vitro synthesis of DNA, that is DNA synthesis of DNA outside the cell, where we can form the DNA, we can form the multiple copies of a particular DNA segment, and we can use for various techniques or various further proceedings. So let us study what is PCR and how it is active. So students, already we have discussed this PCR, the full form of PCR is polymerase chain reaction, and this is mainly in vitro synthesis of DNA for producing various copies, and that is called amplification of the DNA. Now suppose this is the DNA segment and we want this segment to be amplified. That means we want various number of copies of this particular DNA. Clear? Now what to do? First step is the denaturation. So there are three basic steps: denaturation, then annealing, and then extension. So three basic steps are there in the PCR, clear, which are very vital. So first of all is the denaturation. So denaturation, you can, from the name, impart that somehow how it is, we are going to denature this DNA. And what we are going to do it, we are going to do here the breaking the hydrogen bonds, because these two strands are joined by the hydrogen bonds produced by the which are linking the nitrogen bases. So we are going to break this hydrogen bond, and as a result what will happen, these two strands will get separated from each other. Now this denaturation is done in 94 degrees centigrade. Clear? Because at this temperature, the temperature is enough to break the hydrogen bonds. But we can't keep it for a long, because if we keep it for a long at 94 degrees centigrade, it may break the phosphodiester bonds. Clear? And finally we will left with nothing. So for few seconds we keep it in the 94 degrees centigrade to break the hydrogen bonds, and so these hydrogen bonds, you can see here, it is broken down and the two strands get separated, and this is called denaturation. So see children, this is PCR. PCR, the full form is polymerase chain reaction. polymerase chain reaction. It is basically the in vitro, that means something outside the cell, in vitro synthesis of DNA from a given segment of DNA. So that means you are producing numerous copies of DNA from a given segment of DNA, and that is called amplification. So PCR is polymerase chain reaction, which is mainly made for in vitro amplification of the given DNA segment. Now, suppose this is the DNA segment. So, we have to produce numerous copies of this DNA segment outside the cell. So, what to do? So, PCR has three basic steps. Denaturation. The first step is denaturation. Then it is annealing, and finally the last step is the extension. So, denaturation, annealing, and extension. Three are the basic steps of PCR or polymerase chain reaction. Now what to do? First of all, we will heat the DNA to 94 degrees centigrade. So why we are heating the DNA to 94 degrees centigrade? To break the hydrogen bonds. You can see here, there the two strands are joined by the hydrogen bonds, clear, which are linking the nitrogen bases. Now here we will break these hydrogen bonds, and as a result the hydrogen bond between the nitrogen bases will break down and the two strands will get separated. So the main aim of denaturation is to separate the DNA strands, clear, and it is done by heating to 94 degrees centigrade. But it is for a very short duration, because if we keep it for a long time it may break the phosphodiester bonds also, and finally we will left with nothing. So for a very short period, the DNA is heated to 94 degrees centigrade and the hydrogen bond are broken and the two strands get separated. So this is about denaturation. Next step is annealing. Now we all know that DNA replication occurs in 5' to 3' direction. Clear? So here there are two strands, and two strands you can see here. This is 3' to 5'. This strand is running 3' to 5'. So here this complementary DNA will be produced from here only. Clear? Because they are anti-parallel, so it will be having 5' here, and so there will be 3', and this strand is running, this is 3 and this is 5. So complementary DNA will form here only, because this is 3, the complementary, since anti-parallel, it will be the 5 polarity will be here, and so this is the 5 to 3 strands. Now we know the DNA replication starts with a very small segment of RNA, that is oligonucleotide, 2 to few nucleotides of RNA, and this is called primer. So this addition of primer, clear, to this already existing strand of DNA is called annealing, and this is done at a temperature of 56 degrees centigrade. So addition of primer, that is DNA, small stretch of RNA to the existing DNA strand, is called annealing, and that is done in the 56 degrees centigrade. And minutely observe the polarity. Clear? The two strands are running anti-parallel in DNA, and there we are adding in the 3' prime end of DNA, existing DNA, we are adding the new stretch. Clear? Where the new stretch is having 5' to this end and the 3' to this end, and this DNA replication will occur from 5 to 3, so we can extend in this way. Or here it is the 3' to 5', the existing DNA strand, and we are adding the new stretch at this end, 3' end. Clear? Where the new stretch is having 5' polarity to this part, and the 3' polarity to this part, and the DNA replication will occur in 5' to 3' direction. So very minutely observe this polarity, because while you are drawing the diagram, the polarity is very much important. Clear? So carefully locate So mind that the 3' end of the existing strand, new stretch of RNA will be added, and that RNA strands which you are adding should have the 5' polarity to the starting point. So this is adding of RNA. This RNA primer is called annealing, and that is done at the 56 degree centigrade. Now the next part is the extension. So already the 3' end in both the cases are, you can see these are free, and this 3' hydroxyl, we know that 3' end contains the hydroxyl group where the new nucleotides will be added, clear, where the phosphate group will form the phosphoester bond here. So these 3' ends are free in both the cases where these new nucleotides will be added. So you can see here the new 3' end is free where new nucleotides can be added. Here also this 3' end is free, the hydroxyl group where the new nucleotide will be added, forming the with their phosphate group, clear, forming the phosphodiester bond. Now the problem is, and this we know this is done by DNA polymerase. So that's till that part is everything is simple. But so we need lots of oligonucleotides, lots of nucleotides, clear, deoxyribonucleotides, so that the polymerase can occur, and we need DNA polymerase. But the problem is that this is done at a temperature of 72 degrees centigrade, and we know that basically these DNA polymerase or enzymes are proteins, and proteins get denatured or after at this temperature. So normal the proteins which we use in our body or in living system, they get denatured at this temperature. So obviously, so DNA polymerase is also a protein. It is an enzyme which is made up of protein. So obviously the DNA polymerase, if we extract it from any common living organism, that will not work in this temperature. It will get denatured. So what is done? There are certain archaebacteria which grows in hot spring. We know that archaebacteria have a diverse habitat and they can grow in hot spring. So certain archaebacteria are there which can grow in hot spring, which has a temperature of roughly about 100 degrees centigrade. So they have some thermostable, clear proteins, and so this type of one of these archaebacteria is Thermus aquaticus. You can see here Thermus aquaticus. So DNA from this particular DNA polymerase from this particular bacteria has been extracted, clear, and this DNA polymerase, this thermostable DNA polymerase which is there in this bacteria, that is Thermus aquaticus, is called Taq polymerase. T from Thermus aquaticus AQ. So this is Taq polymerase. Clear. This Taq polymerase is used here, clear, to synthesize the new DNA strands. So as a result, we are getting numerous. So this is now became double stranded, and this is also now became double stranded. So now this strand has become double stranded because at this end new nucleotides has been added, and now they extended. This is our DNA polymerase, Taq DNA polymerase causes extension of this strand, and now we have got two double-stranded DNA from a single double-stranded DNA. So likewise, if we repeat this process, we are supposed to get numerous double-stranded DNA from a single double-stranded DNA copy. So this is called polymerase chain reaction, which is mainly meant for amplification of a given DNA segment, and it is of immense importance where the DNA is very meager, and we know that in most cases if we want to go for certain experiments, so we need a number of multiple copies of DNA because these are highly specific experiments. It may went wrong, clear. So This PCR technique is extensively important in any experiments which we are going with the DNA.